So thanks, Dirk, for this lovely introduction, and thank you for this honor to present at CSTAR. I'm really nervous because this is a talk series that I really like, and I watched many of these talks, and I send all my students to watch the talks. And so today I would like to tell you two stories about white matter pathways for language processing in the brain, but we'll start in the beginning. And the beginning was very, very simple. We started off from a very simple model of brain and language, which had only one language pathway, and that was the Arcuate Fasciculus. And in the past few decades, however, we gradually learned that the real picture is different, and it's much more complicated. And in particular, it has many more pathways. And specifically, we started talking about a dorsal pathway and ventral pathways for language processing. And we also added into the picture the right hemisphere. So everything becomes more complicated and more interesting. If we ask ourselves how we got to this position, I would say we first had to realize that linguistic computations like lexical access or phonological awareness and so on rely on multiple brain regions, and the information is processed along multiple converging and diverging pathways. Of course, we knew that already back in the late 19th century. We already had a very clear idea in the literature on neurology of language and the brain that there are converging and diverging pathways for language processing. But then we forgot for a while. And we had to wait until vision scientists kindly reminded us what we already knew once, that the pathways diverge into a dual processing scheme. In the vision science domain, we're talking about what and where pathways. And this inspired models of dual stream in the auditory neurosciences. And it also inspired the dual processing scheme of speech processing. You're all very much familiar with these models, I'm sure. Two of the dual stream models that have been proposed are shown here, one from Hickok and Poppel and the other from Rauschecker and Scott. According to this model, I'm going to move on to a simplified description. According to the dual stream framework of speech processing, we're talking about a dorsal stream that extracts phonological structure and conducts a sensorimotor transform from auditory signals to motor commands, and a ventral stream, bilateral ventral stream, that deals with lexical access and maps sound to meaning. And similarly, in the reading sphere, a similar pathway-based model was proposed by Dahan in his book, Reading in the Brain, and you can recognize easily the same ventral and dorsal streams, with the dorsal stream has access to, providing access to pronunciation and articulation, and the ventral stream provides access to meaning. What I plan to do in this talk is to tell you two stories. The first story is going to be a ventral story, and it's going to focus on the ventral pathways and the role in morphological decomposition, morphological processing. The second story is going to be dealing with the dorsal pathways, and I'm going to tell you about persistent developmental stuttering in the context of the dorsal pathways, and also about sensorimotor synchronization in the context of this dorsal stream. While we do that, I want us to also extend the scheme to some extent to include, particularly in the dorsal stream, other pathways, and specifically the frontal ascending tract bilaterally, and even the cerebellar pathways that I think deserve to be included in the dorsal stream within this story, and I'll explain what I mean. And I'm also going to focus throughout the talk on how we generalize the effects across modality, across languages, across different samples. So that's going to be a recurring theme along this talk. To get started, I want to mention that the methodology we'll use throughout the talk is diffusion MRI. This is probably familiar to most or all of you, but I'm just going to mention very briefly, we acquire images with an MRI scanner sensitized to the speed of water diffusion. And then we quantify the diffusion in many different directions, the diffusion speed in many different directions, and model it with a simple shape of tensor and ellipsoid pointing in the direction of the maximum speed of diffusion. And we decompose this tensor into 3 eigenvalues and calculate various measures to basically get an understanding of how anisotropic the diffusion is in a given point in the brain. So is there a directional preference for water diffusion? And if there is, it's an indication that white matter is probably oriented along this way. So we calculate a shorthand for anisotropy, which is pretty much the standard deviation of the 3 eigenvalues of this tensor. And that's the main measure that we use as a proxy for anisotropic diffusion at any given point in the brain. Other parameters that we might extract from this tensor include mean diffusivity, the mean of the 3 eigenvalues, and more. And the other thing we do constantly is we take these tensors and we follow the direction of the tensors in order to generate streamlines or recreate the white matter pathways, virtual pathways in the brain of each individual that we scan, so that we come up with these beautiful pictures of dorsal pathways and ventral pathways in every individual brain. If we take the same individuals and we have them do some cognitive tests, then we can start doing the interesting things of relating between their cognitive measures on the one hand and the properties of their pathways on the other hand. The other thing we do all the time is we compare between the properties of the pathways between different groups. We compare between the groups along the pathway. So these will be profiles of anisotropy in this case along a given pathway for 2 groups that we would like to compare. We also go along the pathway and we calculate these correlations with cognitive measures to be more specific to a particular spot along the pathway. Okay, so that was my single methodology slide. My second methodology slide is a warning. We should all keep in mind that these diffusivity parameters are affected by multiple structural properties of white matter, not only by myelin, but also by axonal density, by directional coherence, and by axonal thickness. I only now notice that I'm very blurry. Okay. Maybe my camera can hear. All right, here we go. First question that we would like to address is a question about morphological processing. And this was the dissertation of Maya Jablonski, who is here in the audience. What Maya wanted to understand in this beautiful model of the reading pathways, why are we not seeing a role for morphological decomposition? Which pathways are implementing morphological decomposition? Are they the dorsal pathways? Are they the ventral pathways? And of course, when we talk about morphology, I'm not sure how much I need to explain to this crowd. Many people here are probably experts on this. But just to make sure that everybody's on the same page, we refer to morphological decomposition as taking a word and dividing it into its morphemes, its basic building blocks. So a complicated word like representational can be decomposed into 4 different morphemes that are combined to create this word together, to generate this word. And throughout this talk, when I say morphology, I mean derivational morphology. And morphology is a very powerful tool for generating new words and understanding unfamiliar words. And what we wanted to do in this project is to understand where does morphological processing fit in this scheme of the reading pathways. And as always, when you talk about where, we're not only interested in the anatomical location, but also in which type of process are we talking about when we're talking about morphological decomposition. Is it more along the lines of the ventral stream lexical axis processes, or is it more along the lines of the phonological decomposition and access to pronunciation that's carried out by the dorsal stream? So ventral or dorsal. So we needed to develop a behavioral proxy for morphological processing, and the one we chose is the morpheme interference effect. We can talk later about why this measure and not others. And briefly, the morpheme interference effect, and this I'm going to talk about results concerning this effect for a while, so this is a good time to tune in. Morpheme interference effect is the phenomenon that pseudo-words that contain real morphemes in them are harder to reject compared to those that contain invented morphemes. So for example, if you take 2 different pseudo-words and you just carry out lexical decision on them, one of them is going to be harder to reject because it's composed of 2 real morphemes. And the other is going to be easier to reject because one of the morphemes is unreal. There is no such morpheme in English. So we expect people to have a harder time processing these stimuli, the ones that contain only real morphemes, and it's harder to reject them as non-words. And we can quantify how hard it is for each individual in terms of accuracy, in terms of reaction time, to reject these weird pseudo-words. And that's going to be our measure of morphological sensitivity. Interestingly enough, if you do this across many subjects, and this is a project that we carried out in collaboration with Kathy Russell and Joe Taylor at the Royal Holloway, we can see that indeed many individuals find it harder to reject words that contain pseudo-words that contain real morphemes in them, but they also vary to a great extent on how hard it is, how badly they're affected by this manipulation. And some of them are severely affected by the manipulations and others not so much. This is great for us because now we have variability, which is essential to detect correlations with brain measures. So we have to take brain measures, and of course we measure diffusivity in order to quantify properties of white matter in the brains of these individuals. 45 adult English readers were measured by Joe Taylor and Kathy Russell. And we looked at this data and tried to find if there are any associations between the properties of dorsal and ventral tracts identified in every individual's brain and how sensitive they are to morphological structure. And I wouldn't be telling you the story if there was no relation, but happily we found a significant association between the properties of the ventral pathways bilaterally. Specifically, this pathways is known as the inferior frontal occipital fasciculus, and you can see that both in the left hemisphere and in the right hemisphere, it shows a nice correlation. I'm only showing you significant associations that survive correction along the tract, and it correlates with the morphemic cost in terms of accuracy. We also find bilateral ventral associations with the reaction time morphemic cost. You can find this with reaction time and accuracy. You can find it with anisotropy and with mean diffusivity. It's a general phenomenon, but it only generalizes to bilateral ventral tracts. It doesn't generalize to the dorsal stream. So that was the first chapter. And my second chapter of the first story is going to be to attempt to generalize this picture across languages and orthographies. And I want to convince you first that generalizing to Hebrew is far from trivial. So this slide basically says, unlike English, Hebrew is a nonlinear morphological structure. So for example, to combine morphemes in English, you just take the two morphemes and glue them one next to each other. In Hebrew, you have to take a root, a triconsonantal root built up of 3 consonants, and plug it into a pattern in a nonlinear fashion. So Hebrew combines morphemes in a more complicated way. Hebrew is also, unlike English, a morphologically rich language, and it also has a deep orthography, similarly to English. So the combination of both of these together, and specifically the fact that in Hebrew we don't spell out the vowels, suggests that morphological processing actually has a really important role in Hebrew reading and spelling, way beyond the role of the well-known phonological predictors of reading. So perhaps because of this, we know that some hallmark phenomena in visual word recognition do not apply to root-derived Hebrew words. So it's far from trivial that we're able to generalize the behavioral component of what I've shown you so far. And then it's also not trivial that we will generalize the association with the ventral pathways that we've seen for English. So first thing we have to do is generalize the morpheme interference effect to Hebrew. This again was part of Maya's dissertation. And what we created is a paradigm in which, similarly to the English morpheme interference effect, we look at pseudo words only. Both of these are pseudo words. Some of them are derived from invented roots, and others are derived from real roots. And then we're looking for the cost of the real root in the pseudo word. And of course, all these stimuli are matched on similarity to real words. So if we find a difference, it's because of their structural properties, not because some of them are more confusing in terms of similarity. And you'll be happy to know that we can replicate the same effect that you find in English when you look at this phenomenon in Hebrew, such that the accuracy drops and the reaction time goes up for the stimuli that incorporate real roots compared to those that incorporate non-roots. I'm going to skip this. So now we do this in a bunch of subjects, 45 subjects to be precise. And we see, similarly to English, that there is a really nice effect, but also that there is a really nice variability across subjects. As I said earlier, this is really important in order to be able to detect any associations with white matter. Then we collect diffusion imaging in all these participants, this time in Tel Aviv University, and we track the dorsal and the ventral pathways in the left hemisphere and right hemisphere of every individual. And now I'm taking a really long time of working on this data and condensing it into one slide that says we pretty much find similar results in Hebrew to the ones that we found in English. This is a nice way to look at it. In English, you can see the association along the IFOF with the morpheme interference cost. Similarly, in Hebrew, there is an association along the left IFOF. And you can see similar associations in the left inferior longitudinal fasciculus and in the right uncinate fasciculus. So there is a generalization. The details do differ between the languages. And if you want to know the details, there are a ton of them in the paper. I'm not saying everything is exactly the same, but given all of the differences that I just described to you between these two languages, I think it's striking that they give rise to some similarities. And if you take a general look at the data in Hebrew, you can still define it in the same way as we did in English, which is there is a bilateral ventral association with morphological processing measures. So I thought I'm probably not going to have time to get into this part. And this is an experiment that shows how we can get morpheme interference without having people press buttons, just by measuring their microsaccade patterns. But I'm gonna send you to find this paper if you're interested, and keep going so we can get to the second story at some point. So the last chapter of the first story, the ventral story, will look at generalization across task and stimulus modality and response modality. And as I said, generalization is an overarching theme in this particular story. So this is the last chapter in my dissertation, and what you did here was to carry out a simple, nothing simple about this, a similar analysis of pathways, but the task was completely different. So the task in the prior two studies was a morphological task, a lexical decision task that examines the effect of morphology on written words. And participants were simply pushing buttons in response to written pseudo-words that have different structures. In this case, the task had participants listening to real words. So they're listening instead of reading. The stimuli are real words instead of pseudo-words. They're listening to real words, and then it's a fluency task. They basically have to produce as many words as they can in half a minute that share the same root with the word that they listen to. So you have 30 seconds to produce as many words as you can based on extracting the root from the auditorily presented word that you just listened to. As you can see, there are many things that are different between this task and the prior task, except this again relies on extracting a root, and it also requires morphological processing. And when we measure how many words these participants produced, we call this root fluency. We can see that there's a striking correlation between their ability to produce many words and the properties of the bilateral ventral tracts again, the bilateral inferior frontal occipital fasciculus, IFOF, the bilateral ILF, and there are several other pathways showing this effect. Interestingly, you can see I put an asterisk next to generalize, because other than this generalization, we also found an association this time in one dorsal stream pathway, the frontal Aslan tract. And I'll talk more about this pathway in the second story, but it's important to notice here that when you change the task to a task that involves producing language, something changes. And the task really modulates the findings that we can demonstrate in the white matter pathways. So it's not only about the cognitive process that's been carrying out, it's also about the task and the modality that it requires. And there's really, this is a huge paper with two parallel analyses, probabilistic tracking and deterministic tracking, different modeling, and so on. So I'm not even going to try to cover all the details, but they're in the paper if you're interested. So to summarize story one, story one dealt with the bilateral ventral pathways and their role in morphological association, in morphological processing. And we've shown this effect across different tasks, across different modalities, different processing pipelines. Of course, I want to mention that we only tested morphology at the single word level, and you might put words in context and maybe things are going to change to some extent. So that's something that we didn't do and is interesting to explore. The other thing I want to mention is beyond all this generalization, the task and the modality do modulate the association patterns that we find, and that's important to keep in mind. I would say, taken together, this pattern of results supports the view that sensitivity to morphological structure, and more specifically root extraction, is an essential component of lexical access, which is carried out by the bilateral ventral stream. And before I move on to the second story, which is going to be a shorter story, I want to give you a cautionary note. It's tempting to interpret these associations between cognitive measures and tractometry as indicating one-to-one mapping between a tract and a function. And I think this would be a mistake. These are very large white matter tracts, and they do not have a single function. They relay information between distant regions in the brain, and the type of information that they relay might be modality and task dependent. So I think this would be a misinterpretation to frame this as we found the role of the bilateral ventral pathways in morphological processing, but rather we found an association between these pathways and morphological processing, along with other things that these pathways do. And I'm transitioning quickly to my second story that deals with the dorsal pathways and their role in persistent developmental stuttering. And everything we do is going to be in adults. So I'm just going to put this out there right now and forget about the developmental question for a bit. We can talk more later about developmental aspects. So the initial finding with respect to persistent developmental stuttering in adults was a rather simple story. And that story referred to a single hotspot in the brain, the Rolandic operculum, that showed a difference between adults who stutters and controls. And this finding by Zomer, I find very really reminiscent of a finding by Torkel Klingberg in relation to dyslexia around the same time. So dyslexia as well started as a really simple story and then emerged into something much more complicated as time and research went by. And I think it would be nice to have a simple story, but there are a few things we have to consider here. One of them is, this is a voxel by voxel study. When you find a group difference in template space, which is very close to the cortex, there's always a concern of partial voluming. You might be sampling the cortex more in one group compared to the other, particularly if there are volume differences between the groups. And when you look at the details here, if you look at the y-axis, you'll notice that indeed these two groups differ. Actually, anisotropy is very, very low in both groups. And so this raises some questions, whether we can recreate this using different tools. And of course, this was 2002 and many things happened. A lot of research happened since then. But this was really what started us off in this direction. And our approach, as always, has been to track fibers in individual participants in their native space. And then we will try to find our regions of interest in every individual separately and study its properties and try to avoid the problems that I alluded to before. So we recruited, this was close to 10 years ago, we recruited a sample of Hebrew-speaking adults who stutter compared to controls. This was the dissertation of Vered Kornfeld-Duenius. To make a long story short, Vered found reduced volume in the adults who stutter in the left dorsal stream. You can see these differences over here. This is a normalized volume measure. There was no difference in the ventral stream. And then she also found reduced anisotropy in the same group in the right dorsal stream. But again, there was no difference in the ventral stream. And more recently, just this past year, Sivan Yossinger, a PhD student in my lab, has happily reproduced this finding or a similar finding, showing a difference, a significant difference in anisotropy in a completely independent sample collected by Anastasia Sers and Vince Greco in McGill University. So a completely different sample of adults who stutter and controls, small samples similarly to our original sample, which was also 15 and 19. And we find a difference in anisotropy in the dorsal pathway and no differences in the ventral pathway. I'll show you this later. So based on Vered's findings, we came up with a prediction that the frontal Aslan tract might also show a difference between adults who stutter and controls. And the frontal Aslan tract is a pathway that is a fairly recent discovery in the diffusion imaging literature. It started to be discussed in the literature around 2008. In an early paper by Lowe and colleagues, and later on by Catani and others in 2012, there were various papers showing that this tract has something to do with language production and particularly language fluency. Most of the findings were from patients with PPA. Given that this pathway connects between Broca's area and the precentral cortex, and it connects it to the pre-SMA and SMA dorsally, we thought this is a very relevant tract to be looking at in adults who stutter. So Vered developed these automated protocols for identifying the Aslan tract in individuals who stutter at the top row and controls at the bottom row. We did this for everybody in our sample, 15 and 19. And we found that indeed mean diffusivity was significantly different between these two groups in the left and right frontal Aslan tract. There was no difference in FA, and there was no difference in the CST, another pathway we looked at. And even more importantly, if you look at a certain part of this pathway that shows a significant difference between the groups, we could also show that adults who stutter show an association in this segment with speech rate, which is not found in the control group. And again, in a recent reproduction, Sivan has found a similar pattern of results in an independent sample from McGill. There is a significant difference in mean diffusivity. It's elevated similarly to our original findings, elevated mean diffusivity in the frontal Aslan tract. And you can find this in the left and in the right frontal Aslan tract. The details vary a little bit, but this is again sort of a replication across languages, across samples. In this new sample as well, there were no differences in the ventral pathways at all. The last part that I want to talk about is a project about the cerebellar pathways of adults who stutter. And it started with, again, developing tools for looking at these pathways. And once we had these automatic tools to segment these pathways in every individual participant, we could start looking at the role of the cerebellar pathways in persistent developmental stuttering. So Sivan has carried out these segmentations in every individual. These are participants with persistent developmental stuttering, and these are control participants. And this is the same sample that we looked at before, 15 and 19. There were no group differences in the cerebellar peduncles of these participants. We're talking about the superior, the middle, and the inferior cerebellar peduncles. You can see how perfectly the profiles along the pathways align. This is a perfect test-retest validation, if you ask me, but no significant differences between the groups. However, if you look at the association along the pathways with speech rate, in the left inferior cerebellar peduncles, there is a significant association in the stuttering group, and there is no association whatsoever in the control group. And this difference in associations was significant. So this is just out now in brain structure and function. And I think this is an interesting finding, and we'll discuss it in a minute. This is a similar flavored finding that I want to discuss in this regards, but this is a different experiment. So in this data that we are working on in collaboration with Anastasia Sears and Vince Greco, Anastasia also collected data on a behavioral sensorimotor synchronization task. Individuals are basically listening to a metronome beat, and they have to synchronize their tapping to the auditory beat of the metronome. And if you do that, if all of us would have done such a task, we were all going to tap before the beat of the metronome. And as you can see, if you measure asynchrony is always negative. So all of us would tap before the beat of the metronome, or nearly all of us. This is a control group, and you can see that they're negative. So they show negative asynchrony. It's a standard result. You can already see that the adults who stutter are more negative. And there is a paper on that Anastasia published already. But in this analysis, we wanted to look at this asynchrony level in association with properties of the cerebellar pathways. And indeed, the level to which you are asynchronous with the auditory beat is associated with properties of the left ICP. Again, same pathway. This association was only found in the group of adults who stutter. It was not found in the control group. And the last bit of the story that I want to tell you about is if you look at the Arcuate Fasciculus, and we had a pretty good reason to look at the Arcuate Fasciculus because in a previous paper just in control participants, we found an association in the Arcuate Fasciculus with asynchrony. So we went to look at recently went to look at the Arcuate Fasciculus and analyze the association in the Arcuate with the level of asynchrony. And it's interesting that the adults who stutter do not show any association in the Arcuate, but the controls did to some extent. And so this is somewhat a reproduction. The effects are very weak. This is a small sample of controls, but you can see something reminiscent of what we found in just another sample of controls. And you do not see the same level of association in the adults who stutter in the other group. And this difference is pretty significant. So two things that I want to say about this. First, I want to summarize and say we've looked at stuttering related differences, and we found them only in the dorsal stream. We did not find any differences in the ventral stream. We found differences in the frontal ascending tract and in the cerebellar peduncles as well. And I would like to say maybe we should think about these pathways as part of the dorsal stream, given their role in sensorimotor synchronization and integration. I already want to say, of course, when we're looking at adults, we are not in the business of saying anything about causality. There's nothing we can say about what caused what when we look at adults who stutter versus adult controls. So it's very possible that some of the differences which we find in association patterns, in diffusivity properties, they might be caused by years of living with stuttering, or they might have something to do with the etiology of stuttering. At this point, we cannot dissociate between the two. I do want to make some interesting predictions, and then I'm going to close. I think, and this is rather speculative, but I think there is a potential prediction that sensorimotor synchronization training might cause changes in the dorsal and cerebellar tracts. And if that happens, how amazing would it be if these changes are also fluency enhancing? Now, of course, as I said before, this is a classic mistake to think that if you find two things in the same pathway, it means that they are the same. So I'm not saying that, but I am saying maybe this is not just a coincidence that we find these associations in the dorsal pathways, and maybe if we train, we can induce change, and then we can know for sure if they are causally associated or not. So this is something we can do, and I'm pitching it out there because training studies are hard, and we need many people trying and pushing the limits of perturbation to the system in order to get closer to causality. The last point I want to make is we've seen that association patterns between tractometry and cognitive measures sometimes reveal differences that were invisible when we only compared tractometry between the groups. And I'm talking about this finding. And this is not the only place where we see this. In fact, in the past, when we looked at prematurely born children compared to full term, we've seen some similar differences in association patterns, even in tracts that did not show a significant difference in their diffusivities. And I found similar findings between high and low performing preschoolers in a recent paper by from the Federici lab. And I found a similar finding in a recent paper from the PI lab with people with primary progressive aphasia versus controls. Not in every pathway. Many pathways are different between these groups, but in the IFOF specifically, no difference between the groups. But if you look at the association patterns, they're different. So I think this is a more general phenomenon, and it might show us that associations are more sensitive, and we can gain a lot of sensitivity by looking at associations beyond group differences. And potentially this means that there is a differential reliance on a specific track for a given cognitive process by the two groups. That's how I would interpret this. So I want to just thank many collaborators on these projects, and specifically, I talked a lot about projects by Maya and Sivan, and so I want to thank them both. And thank you. Dirk, I hope your Zoom is working now.
Yes, well, so thank you so much, Michal, for your wonderful talk. That was great. And I'm happy to moderate the questions in the chat, but I can only see the ones that came in after I logged in. So if you don't mind, Michal, if you can read out the first questions that you got, and then I can take over once we get to the questions from them.
Okay. So I can see a question from Will Hula saying the associations between FA and the morphological lexical decision task look to be both positive and negative at different local points along the IFOF. Is this a correct reading? And if so, can you comment on these patterns and what they might mean? So let's talk about, I'm taking this more generally as, let's talk about direction of association. Because we didn't have a case where along the same pathway there is an alternation between positive and negative associations, but in different pathways, looking at different cognitive measures, sometimes there are negative associations, sometimes they're positive. This can happen with when you switch from anisotropy to mean diffusivity, of course, and when you switch from reaction time measure to an accuracy measure. But no matter how you do it, I would say that the direction of association is not something that we can really rely on when we interpret these findings. So in the past, and this is not specific to these findings in reading as well, we've seen negative associations with anisotropy in many different pathways. They go against the common logic only if you assume that anisotropy equals myelin. But given that we know that anisotropy is affected by many different factors, I think we should let go of this idea that there's something better about positive associations compared to negative. We can detect associations that tell us something about the role of this pathway or the relation between this pathway and a given function. And later on, there's a lot that we can do in order to further explore these findings. For example, we can use quantitative MR measures. Sometimes those are more specific. Some QMR measures are more specific to myelin, for example, quantitative T1. So if we decompose the finding into different quantitative MR measures, that could tell us something about the generators of the effect. Direction of association is not something I would rely on in interpretation. And we have such a finding, maybe I'll mention it later. Okay, Dirk, the second one is from you. It says, when subjects accept Towerly, does that mean they are wrong or that they allow for the productive use of the suffix? And is this relevant to the interpretation of related functional activation? And do you want to unpack this a little bit for me, or should I just shoot?
Well, it gives me an advantage over other question askers, right?
Everybody can unmute and yeah. When I discuss their question democratically.
Well, I mean, so it seemed to me the way you were presenting it is that if you accept that as a word, that's an error. And to me, I'm not sure if it is an error. It means that you allow for, I can imagine a word towerly that's like a tower. It means like mm-hmm. a tower, so it's not a problem. Mm-hmm. Mm-hmm. So I was just, so if you then equate that with accepting tower la, which really is an error because that is nowhere near a real word, it seems to me that you're looking at two different processes. One is an inability to identify a non-word, and the other one is use of morphemes in a productive way.
So basically, what I think what stems from what you're saying is perhaps those who are making more mistakes in this task are those who are using morphology in a more productive way. So they are the higher morphology participants, the ones who are making more mistakes. And that's very possible, actually, quite possible that those who have higher costs are using the morphological generator more than those who have lower cost. That's fine. It's yet another reason why I wouldn't interpret the direction of association in one way or the other, because it's not completely clear in this task what is better. So that's what I would make of it. I would say that the fact that the reaction time, when you see a cost in reaction time, I think it's, well, I'm not sure if it tells the same story of the mistakes that you were just talking about. But there are a bunch of things that we could do in order to look at this. We could partial out their reading ability. These effects survive when you partial out reading speed, for example, so being able to recognize words really quickly, things like that. So there are a bunch of things you can do in order to try to understand the morpheme interference effect better. But it's very likely that it reflects higher reliance on morphological generators. I'm okay with that. Next question is from Francesco Tomaiolo. The diagram you've shown at the beginning presents several steps along with the dorsal and ventral network. On the contrary, the DTI shows a single line that goes from occipital to frontal regions. May we say that we're looking at several synaptic stops and say that what we see is a stream rather than a single fascicle? I would say definitely. DTI fiber tracking and even probabilistic fiber tracking with the fanciest modeling scheme of constrained spherical deconvolution is always likely to jump synapses, because within a voxel, you're modeling a whole population. So you're modeling diffusivity in a box, and it's affected by axons going in many different directions. And it's completely possible that the general population signal will lead you to continue tracking even though some of these axons stopped. Specifically regarding what you're commenting on, there's been a discussion about the IFOF specifically, whether this is a real pathway or maybe it makes a stop along the way. I've sometimes shown only the IFOF, but in many cases, what we see is there are associations in several ventral pathways, not only in the IFOF. So I'm not making any particular claims about the IFOF. Sometimes the associations are found in the inferior longitudinal fasciculus, in the uncinate fasciculus. It's very likely that they concern several different pathways in the ventral stream, and not specifically the direct connection between the occipital and the frontal lobe, if it exists. So I hope this answered the question, but if not, just let me know.
I can now see the rest of the questions too, so I can let you off the hook a little bit, Michal.
Okay.
So the next question is from Danielle Fahey, who is a postdoc right here at USC. For the rejection of pseudo words with real morphemes, which types of morphemes are included? For example, both derivations or inflections with lexical morphemes. Also, are all participants in the studies you describe about morphological sensitivity monolingual speakers?
Okay, so two questions. For the first one, we only use derivational morphology and manipulate derivational morphology in these studies. We so far did not look at inflectional morphology at all. And so the answer is only derivational morphemes. I'm not sure. Well, I don't want to get into more detail about that. But the simple answer is only derivational morphemes. And when it was a Hebrew manipulation, as you've seen, we manipulated the root morpheme, which is of course derivational. In the last study that I've shown with morphological fluency, we manipulated separately the morphological root and the morphological pattern, but both of them are again derivational morphemes. And in regards to the second question, are all participants monolingual speakers? In Israel, you cannot really find monolingual speakers, I think, or you have to work really, really hard in order to find adults who do not know English at all. And so I would say this might be a difference. We don't know exactly how many languages are spoken by the English readers, but I can bet that the Hebrew readers are probably all bilingual with English to some extent, and very likely that some of them are more than bilingual. So there's probably a difference in these populations.
Great, thanks. The next question is from Julius Fredrickson. The bilaterality of your findings is striking. Do you think this means that morphology relies on both hemispheres, or do the right hemisphere results just reflect the fact that density of homologous white matter tracts is highly correlated?
This is an interesting idea. We definitely thought about the first one. So my understanding of this effect so far has always been that because we find it so repeatedly, and because we do not find all effects to be bilateral, only ventral effects, primarily, not only, primarily ventral effects to be so bilateral, my interpretation was that both the left and the right pathways are involved in processing this information. This also fits with the idea that started in the dual stream model completely independently, that the ventral stream is more bilateral and relies on the temporal lobes in both hemispheres. However, what you're suggesting is, and we can test what you're suggesting, maybe we have, and there are a lot of analyses that have been done and I don't remember all of them, but it's possible to test, it's possible to partial out the internal associations between the left and right homologous pathways and see if the association survives this. And do this twice, once for the left association and once for the right association. Maybe Maya remembers if we tried that, but I don't recall. We can definitely test. My hunch is it's not going to change the effect. My hunch is if the data is strong enough, we would be able to see that the association is not due to the intercorrelations between the hemispheres. I'm trying to think if there's more data from other directions to support this bilaterality. And at the moment, this is what I'm going to say about this.
Thank you. We have a question from Tatjana Schnur. The analysis sometimes use cluster FA, cluster mean MD, or just FA and MD. Why did you make these different choices regarding tract integrity measurements, and what does it mean to find different results across the different variables?
Very good question. I think we're just trying our best to find interesting associations between properties of white matter and properties of cognition. And it's quite striking. I mean, once you start seeing these things over and over, you get used to it. But if you think about this, these are two, or not two, these are several independent measures that you're taking from the same individual. Well, not independent, but definitely different measures from the same individual. It's not trivial that you're going to find these associations across a group. And so I think we do what every cognitive neuroscientist which is you define ahead of time a few different parameters that you're going to look at: reaction time and accuracy, or bold signals in a few different regions of interest, things of that sort. And then you do the analysis, and whenever you find something that looks like a phenomenon, you start testing it in many different directions to make sure that it's not just a quirk. When you start seeing in the same pathway a correlation with mean diffusivity, a difference between two groups that vary on a related measure in mean diffusivity, you start building a little story around it. What I would say is generally our approach has been, I mean, it might have sounded like we're just fishing all over, which is not what we're doing. Generally, our approach is we go along the pathway, we correct for multiple comparisons along the pathway. If we found a tract, if we find a significant cluster, we extract the data, anisotropy or mean diffusivity from that, and we look at, we show the association in a scatter plot. This might generate the feeling as if we run our statistics on the scatter plot, but we don't. We run the statistics along the pathway, correct along the pathway, and then we just show the scatter. This is also why I never show the R and P values across the stock on all the scatters that I've shown, because the R and P values are actually in the calculation along the pathway. So there's no point in recalculating them only on the cluster, and that would be a mistake. My point in answer to your good question is we look specifically at FA and MD. We look along the pathway, and in if we can do less comparisons, we prefer it. So in some cases, if there is an association just by looking at the entire tract altogether, this is one comparison compared to many. So this is preferable.
Great, thank you very much.
Tatiana did that, I was talking for quite a while, but I hope I answered this.
I guess I had one. I mean, a priori, do you think you're getting different kinds of information if you find, like you did towards the end of your talk, you find results with the mean diffusivity, but you don't find it with the FA value? Is that telling you something
right
inherently different, or is it just that, like you mentioned, by chance maybe you either didn't find it in the first case or you found it in the second case?
So no, it's pretty consistent. So I wouldn't think it's by chance, but I'm glad you followed up, because one explanation, there have been papers showing just based on simulations that mean diffusivity is a more sensitive measure. And so in many cases, you might not have found an effect with FA because maybe the sample size was marginal. And then you might see the effect in the mean diffusivity because that's a higher, mean diffusivity has higher power, just more statistical power to the measure. So that would be one explanation why you sometimes find it where you don't see a significant anisotropy effect. That paper by DeSantis and colleagues also shows that they're not, mean diffusivity and anisotropy are not perfectly anti-correlated. They're not just two sides of the same coin. And so it's very possible that one of them shows an effect and the other doesn't. And I really think about this similarly to reaction time and accuracy. For example, sometimes you'll find the effect on reaction time and it will not translate into accuracy effect, even if you're not at ceiling. And that's just the data.
The next question is from Suun Chang. Previous studies of adults who stutter have reported FA differences in the cerebellar peduncles. How do you reconcile these findings with your results looking at the CPs? Also, how are you interpreting the ICP findings in relation to stuttering persistence/severity?
Okay, so yes, when we started looking at the cerebellum, we definitely were already aware of the findings from Connolly and Watkins, showing significant differences in the cerebellar peduncles between adults who stutter and controls. And we could not replicate it. We tried over and over, including, so initially we conducted completely different analysis compared to the ones that they did. And so the first thing we said, okay, so different analysis, maybe that's the reason. So we went and repeated the exact protocol that they carried out on their data, and we still couldn't replicate the differences in the cerebellar peduncles. Even when we defined this was a vital, this list set and defined these regions of interest manually on every individual, and still couldn't replicate the effect. Okay, so there are a bunch of different reasons why somebody doesn't replicate somebody else's effect. One is these are different samples. This is not very convincing. The other is in Connolly and Watkins, the age range was very different than in our study. And actually, there's even a beginning of a finding that shows that the effect changes with age. Our age range was much larger, and it's very possible that we don't find the effect because we're not sampling the process that goes on in individuals' brains at the same point in time. So it's possible that if we had a similar sized sample in the similar age range as the one in Connolly and Watkins's paper, maybe we would see the same finding. I can't say. Our sample was not large enough, so we can start dividing it into the younger ones to fit better with the other sample and the older ones that do not fit. So this is how I would probably attempt to explain it. Hopefully it's a matter of age or it's a matter of variability. And as you well know, in adults who stutter, there's tons of variability, and it's always possible that we didn't find it because the samples are just different, that we're sampling different subpopulations. So it's in answer to the second question, how am I interpreting the ICP finding in relation to stuttering persistence or severity? Which findings in particular?
Hi, Michelle.
Hi, Sion.
Can you hear me?
Yes.
Hi. So the right ICP, I think you found differences in it was not a group effect, but you found some relationships with was it the speech rate results? So
left ICP and speech rate, yes.
I see. Okay, so it was the left ICP. So you didn't look at that in relation to stuttering severity at all. And, by definition, because these are adults, these are persistent stuttering.
Okay. I got this now. Okay, so I'm not going to be able to find this probably. Yes, they differ on speech rate, of course, as you suggest. Because these are adults who stutter, and of course their speech rate is slightly lower than the controls, and there's also an association with severity that you can see when you look at speech rate in relation to the percent SLD. And now if you take this back into the question about the association with the left ICP, what happens if you look at the left ICP with relation to speech rate, and then you partial out SLD? Is that the question?
Yeah, I think that was my question. Mm-hmm.
It's definitely in the paper. I cannot tell you the answer because I don't remember, but it's definitely something that we did. So I'd have to check in the paper. We can both do that.
Thank you so much.
All right.
On the age differences between your samples, the next question is highly relevant from Emily Garnett. What might your hypothesis be regarding similar or different findings in children who stutter?
Oh, okay. Excellent question. If I had to guess, I would hypothesize that the cerebellar differences that we see, or the cerebellar associations that we see, are something that emerges fairly early. With respect to, but this is, this is just my general thoughts about which differences that we see in adults reflect coping strategies, and which ones are more directly related to the etiology. And my speculative intuition is that the cerebellar findings could be traced to an earlier phase and are part of the etiology, whereas what we see in other parts of the brain, including, for example, a whole study that we did on the corpus callosum, frontal callosal connections. Over there, I think we're definitely talking about something that emerges later in life and is an attempt at recovery or compensation. Sorry, I lost the word. So that, but really, we haven't done anything with children, and the literature has many findings, and actually we can both turn to Suyun to say her opinion on that. I think multiple findings in children. I don't think we're going to nail it down to a single factor. We're probably going to have to live with the fact that it's a multifactorial phenomenon.
Okay, and Emily is right there, so she is in touch with that work.
Yeah. Oh, yeah. I forgot. Of course.
The last question for today is from Junyua Ding. Great talk. It is common some tracts cannot be found in some subjects, such as right AF, frontal Aslan tract. How do you deal with those subjects?
Okay, so yes, that's true. In some participants, you cannot find some of the pathways, and specifically the right arcuate. This is true, especially if you run deterministic tractography, the right arcuate. If you run probabilistic tractography, as we've shown even at 2011, you can find the right arcuate. Sorry, the right arcuate. In all participants, pretty much. So probabilistic is not that sensitive to that problem, but there are other problems in probabilistic tractography. In general, what we do is we only analyze the participants where we can find the fibers, and this is yet another reason why probabilistic tractography is an advantage. However, when you run probabilistic tractography, you can find these pathways, but there are many pathways that look completely crazy. So you must start finding ways to select between the fibers that are real and the fibers that are errors, or more likely based on the data and less likely based on the data. So you run into different problems. I would say in general, there's usually enough participants in every group where we can find the fibers, and then we can analyze those. And sometimes the right arcuate is causing us problems in this regard. In the frontal Aslan tract, in many cases, we're doing just fine. It's a pretty robust pathway. So we usually don't have to deal with that problem over there.
Great. Thank you so much, Michal, for your wonderful talk and for the way in which you've answered all of the questions that we had in the chat box. Thanks for joining us, audience. Thanks for your questions, and we hope to see you again in 2 weeks' time, when we'll be listening to Davida Fromm and Brian McWhinney on the Aphasia Bank. Good day.
Thank you.